Hi there! Today I will be talking to you about my final project, and I will give you a visual representation of my undergraduate research. My undergraduate research is called Cloning of the RII1 Promoter, and this is found in humans. So I'm going to start by giving you a little bit of a background, if I may. So RII1 stands for retinoic acid-induced 1. This is a gene. And this gene is located on a chromosome 17 on the petite arm. As you can see here, I've drawn out a chromosome right here, and it would be on the shorter arm. That would be the petite arm. And what we know about RII1 is very little, actually. So we don't know much about the function, and that's part of the reason why I need to clone it. So once I've cloned it, I'll have the sequence, and then from the sequence I can figure out what types of things are known to bind to it, and then from those things I'll know the function. So that is basically my objective, is to clone the promoter so I can find out more about the function. And I'd like to thank Dr. Zees for being my research advisor for the past couple of years, because without her this would not be possible. So let's get into the research portion of this. So first we have to create these things called phosmids. Phosmids are what hold DNA. You can't just take DNA from, say, your mouth and just be like, okay, I'm going to run a research experiment with this. There's a lot that goes into it before. So first you would take DNA and you would shear it, and you would basically break it apart and then put it back together after it's been purified, and then you would package it into these tiny little picture things that you see here. And then you want to culture it, grow it in a culture, and the yellow plate right there symbolizes bacteria, a bacteria plate that will grow overnight and basically reproduce multiple copies of our DNA. And we use bacteria because they're really fast at reproducing. Basically, you can get more of your DNA overnight, and that's called an overnight culture. After I have that, I'm going to go ahead and save all the DNA that I have in this little microcentrifuge tube that's seen over there. Next, we're going to isolate the DNA that we just saved. So I have 5 phosmids. So I have 5 microcentrifuge tubes, and the phosmid names are M17, N24, G12, G18, and J12. And what I want to do is separate the DNA from the liquid, essentially. I mean, none of this stuff is very visible to the eye, but there's a lot of chemical reactions that will be run through in order to isolate the DNA. And the protocol that's used is called a MIDI prep, and that basically washes the DNA and purifies the DNA so you have the purest form to use for future research. And this actually, because phosmids are so big compared to regular DNA sizes, this takes quite some time. Usually I'll have to dedicate 5 to 6 hours after to get this done. So next we're gonna talk about a restriction digest. And a restriction digest is basically where you take one of these phosmids, that's a circular thing that you see right there, because all DNA is usually in a circular form, and I'm going to cut it up. And the way I'm going to cut this up is DNA has these sites where enzymes can bind and slice them, and this is what a restriction digest is. It slices the DNA. And then I'm going to run it on a gel, and basically what the gel does, it separates those fragments that I just cut up, smallest to largest, and as you can see, that's what is on that gray box, which is a gel. This is called gel electrophoresis, and I'll do this for all of my phosmids, and that's why you see different lanes. And you can use different enzymes, as I did in my research experiment. Some of these are called BAMH1, KPN1, XBEL1, and these basically are used also to cut wherever you would like. Next, we have the DNA transfer, and that's basically taking the small fragments that we have on the gel electrophoresis gel that I just showed you and transferring the DNA onto this nylon membrane. And so it's going to move upward, and I'll show you how that's. So basically what we have on our left is our agarose gel that we were just looking at earlier, and in order to get the DNA from the gel onto the nylon membrane, which is where the membrane, it's labeled membrane, you have to go through a southern blot. What a southern blot basically is this apparatus that you set up, and it has the gel laying, and then it has the membrane on top of it, ton of paper towels, and this buffer solution that allows the DNA to move upward through capillary action and it moves on to the nylon membrane, and this can happen overnight. So it's a really easy setup. It's basically just DNA moving from one gel onto an actual sheet of paper, which I'll be able to use for the rest of the experiment because the DNA binds to it easier. It's more stable to use through washes and chemical reactions later on. So that's what we're going to do next. So now we have hybridization, and just to give you a brief explanation of this, I want to remind you guys that the RAI1 gene, drawn right there, has the promoter upstream, and then it has these things called exons, which are just regions within the gene that we want to look at. And so let's get into hybridization. So basically what's happening here is that we've moved our DNA through our southern blot onto this nylon membrane, and that's the gray sheet that you see right there. It's very light, and the reason why the DNA fragments look so faint is because you can't see them to the naked eye. And so we have to go through a process of hybridization and detection in order to be able to see this. Basically what we're going to do is take a promoter that's going to bind to, I'm sorry, we're not going to take a promoter. We're going to take a probe that's going to bind to the promoter, and that we'll be able to later detect later on, and that probe will also be complementary to where the promoter is. So that's what region that we're looking to find on this nylon membrane, but now we have to detect it first in order to see it through a series of washes. So we're going to have an antibody bind to it in order for us to see it. So let's go ahead and talk about detection. So for detection, we're just basically finding the DNA fragments where the probe also binds to, so we can clone it. So let me tell you a little bit about the process now. It's pretty short. And so this is what detection basically is. We have this nylon membrane, and where the orange is represents where the probe has bound, and it's not visible to the human eye, and we need to be able to see it to tell which fragments the probe can bind to, so we can clone just those fragments or those pieces of DNA. So we have to go through a series of washes. First we'll go ahead and use a washing buffer to wash the probe off, and then we want to put blocking buffer on, so make sure nothing else binds to the probe to give us any false positive results. And then we will take this antibody that will also bind to where the probe has bound. So the antibody will be where you see the orange lines. And then we'll go ahead and wash the antibody off. And then we'll put detection buffer on there. And it will basically light up where the probe was. And we will be able to tell which fragments have RAI1 and the promoter of RAI1. So then what we can do is go back and take, say, M17 if it lighted up, or G12, and go ahead and send it for it to be sequenced and cloned, and then we'll have our final results. So basically what we were trying to do is find the plasmids that have the gene RAI1. So let's talk about the results. From my research, I found that M17 and N24, there was no detection, so that means the sample of DNA that I had did not have RAI1, so hence the big X's. And then, so that basically tells us it cannot be cloned, whereas my other samples of DNA, other 3 samples, did have RAI1, so they can be cloned, and they will be sent for sequencing. And then that gives us 3 successful clone copies of the promoter of RAI1. And that's basically what we wanted to begin with, and it was pretty simple to explain, I guess, but it did take pretty much a year and a half, and it will be continued by Dr. Zeese in the future, or her other future students, whoever would like to continue it. We can have repeated trials or further investigation of the human genome sequence. So that's all. I hope you guys enjoyed this and got a lot out of it.